Hello, everyone. I hope everyone is having a good evening and welcome to Oxford Biomedica's webinar about cell and gene therapy. Remember, this session is recorded, so if you do miss anything, don't worry. It'll be uploaded to our YouTube and our website within the coming days. And if you do have any questions throughout the webinar, you can leave them in the chat and the Q&A box just off to the right there. So, without further ado, I hope you're ready to get learning and gain some lovely insight with Oxford Biomedica. So I'll let them commence with their session.
Hi, good afternoon, good evening, everyone. So I'm James Miskin. I'm the Chief Technical Officer at Oxford Biomedica. We're just going to run through a little bit of background around our ABViP collaborative training partnership together with our partners at the University of Oxford and UCL. So next slide, please. So the way this presentation will work is I'll give a bit of background to the CTP itself, to the whole cohort training and experience process. Then Professor Kasim Rafiq from UCL will give a quick overview of the UCL role in ABViP. And then we're hoping that we're going to be joined by our colleague, Professor Kath Green from the University of Oxford. She's literally on getting off a train any second. If that doesn't work, I'll have to do that a little bit. And then we have a run through of some pre-recorded videos for each of the projects that are planned for 2024 cohort entry. We acknowledge these are pre-recorded. Some of the sound on them isn't really loud, but you will have access to the pre-recorded session afterwards. And then we'll open to, after a little bit of an overview on the application requirements process, we'll open for Q&A. The next slide, please. Next slide. So BBSRC is the UK's Biological Research Council, and essentially they funded a number of collaborative training partnerships. Ours is the only one in this specific sector. So in total, there's £22.5 million worth of funding from UKRI through BBSRC into this scheme, which is shared across 10 different CTPs funded by different organisations. So the specific aspect of CTP is that it's a doctoral training program, but it's led by industry but partnered with academia. And each of the programs range between 15 and 35 students per program. And I'll give a bit of background as to the size and scale of Oxford Biomedica's and in a sec. Next slide. So the aim of the CTP is to address the fundamental and applied bioscience challenges associated with viral vectors for future gene therapies. So that's quite a broad title. What we essentially have identified is projects that are specifically designed to understand the bioscience of viruses, not necessarily having to have a formally commercial means of exploitation at the end. So the focus is on biological science and fundamental research. The key focus is to provide the student with a really solid training in this sector, but also with the advantage of it being across both academia and industry. So you get to see a lot of both aspects of this space. So overall, it's a 7-year program with a total of 24 students, aimed at 3 years intake of 8 each in 22, 23, and obviously what we're talking about is the projects for 2024. Each of the projects is fully funded with funding from ourselves, the BBSRC, and each of the universities are putting in funding for several of the students as well in total. And the idea is to create a research hub and increase the breadth and capacity of research. And to bring biotech and academia even closer together. Next slide. So this is just an overview of the management board. I'm not going to read through it. So I'm the CTP director at Oxford Biomedica. If you look at the makeup of the board, essentially we have 3 representatives from each of our partner organisations. So 3 of us from Oxford Biomedica, 3 from UCL, and 3 from Oxford. And on the call today, we're joined by Kasim Rafiq and Catherine Green, we hope. And we also have my colleague Kiri Mitrofanos, who's our Chief Scientific Officer, and the Programme Manager Neelam, who's sharing her presentation at the moment. So next slide. So the overview of research areas addressed by the CTP are quite broad, in and around viral vector design, virology and vaccine development, around viral vector technology development, cell line optimization, process development, analytics, etc. But also around formulation, bioprocessing, and those support areas. Because it's funded by the BBSRC, the underlying feature of each of the projects is that it has to be blue sky biological research. So it can't be too on the engineering spectrum, and it also can't be too on the clinical or preclinical development spectrum. So hopefully that gives you a bit of a rough idea. And there's all sorts of areas that we're focusing on around viral vector, vaccine design and engineering, using data science, and there's a few data science based projects that we're funding across the 3 years of intake, including analytics. And the idea is to really improve viral products for future gene therapies and vaccines. Next slide. So a little bit about the cohort experience and training. Next slide. So part of the thesis of the CTPs is that you provide this really strong cohort experience. That cohort experience comes in with a number of different key features. So there's the obvious things around cohort building. So we have some specific events where the cohorts all get together. For instance, back in the summer, we ran our first symposium, and we'll have one of those in each of the summers. Next year, we'll have both cohorts 1 and 2. This year, it was only cohort 1. In terms of outreach, we'll have a number of different activities around attending STEM events and trying to reach out to the wider sector. We're very, very keen on making our students extremely employable. And we think it's a particularly unique advantage of a CTP in that you get the visibility and direct experience of working both in an academic and an industrial setting. So that's extremely good in relation to one CV. And of course, knowledge. So a PhD or a DPhil is absolutely about training in science and understanding how to do good research. So all of those features are there. And if you look in the list, there's a number of different particular features that are specific to this particular CTP. Next slide, please. So the student experience and supervision support structure is well thought out as well. So each individual student will have a primary supervisor at the host organisation as well as a secondary supervisor. We also provide an industrial supervisor at Oxford Biomedica. We then provide support and mentorship, and the program manager, Neelam, who's, as I say, on this call today, provides additional support and is our primary point of contact for students. It is a student-centered program above and beyond what we would expect from a typical PhD training program. And we're aiming at attracting the highest caliber students. And certainly if the first and second cohort years of intake are anything to go by, we've succeeded in achieving that. We target interactions with external doctoral training programs. Both Oxford University and UCL have other doctoral training programs that we're able to leverage from that perspective. We aim to recruit from a wide pool of students, taking into account a significant amount of, making sure focused on equality and diversity and inclusivity. That experience gained at the interface between academia and industry is something I personally feel extremely motivated by. It's the way to improve the sector by having people who are able to navigate across both facets, and obviously creating a network. So next slide, please. Cohort experience does give that unique interaction between academia and industry. There's an industrial placement that each project will include, and that will include a minimum of 3 months spent at Oxford Biomedica. In some projects, it's actually substantially more than that. Placements will be undertaken that are in relation to that research project and should be developed in collaboration with all the supervisors to help interaction across the cohort and between cohorts. The training for the students will include some quite broad academic training modules that are specific, but also more general ones as well. And then a broad training for students in relation to wider business things such as intellectual property, project management, and things of that nature. Next slide, please. So I'm going to hand over to Professor Kathima Rafiq from UCL. Next slide.
Hi, good evening, everyone. Many thanks, James, for that introduction to the program. So I'm now going to give a brief overview about the activity at UCL. So I'm a professor in cell and gene therapy bioprocessing, and I'll be giving a quick overview about some of the activity we have at UCL and how the program will link within the faculties that we have. Next slide, please. So, just to kind of quickly outline about UCL. So UCL has been leading in a number of areas and is the first in a number of areas. It was the first university in the UK to admit individuals from any background, regardless of their race, religion, social class, and gender. So we were the first university to admit individuals from different religious backgrounds, as well as females as well. The first to teach in areas such as English, engineering, German, and chemistry. And more recently, the first to start its own open access university press. Next slide, please. We also have a world-class research and teaching activity. We are the largest university in the UK. And we're second in the UK for research power. And I think as part of ABViP, you're very fortunate that you've got the first, which is Oxford University, and the second, which is UCL in terms of research activity. As I mentioned, we're the largest university in the UK with over 51,000 students, but that's supported by over 16,000 staff across 11 different faculties. Next slide, please. Talking about the faculties, we have, particularly with this ABViP cohort this year, we have projects, 4 of the projects which UCL are offering spread or span across 3 different faculties. So we have a project that is being offered by biochemical engineering, which is part of the engineering sciences faculty. We have 2 projects being offered by the Cancer Institute, which is part of the Medical Sciences faculty. And we also have a project being offered by the Wolfson Biomedical Research Institute, which is part of the Faculty of Life Sciences. Next slide, please. UCL is at the heart of London, and we're very fortunate to have both an active research environment in one of the world's leading capitals. As you can see here, we're positioned right at the heart of London, very close to Euston Station and King's Cross St Pancras, as well as the British Museum and Great Ormond Street Hospital, which is part of UCL and is a key part of our gene therapy research activity. Next slide, please. Talking about the ATMP and gene therapy activity, UCL is clearly one of the leading UK universities within this space, where we're running the most clinical trials within the ATMP sector, the advanced therapy medicinal product sector. And that includes cell and gene therapies, and it includes CAR-Ts, and some of the projects being offered this year involve investigators who are leading some of those clinical trials. Next slide, please. And then finally, just to highlight on some of the ATMP translation. So not only is UCL actively involved in running a number of clinical trials involved with ATMP development and translation, but we also have a large research funding activity, over £165 million of research funding within the sector. And as you can see, we've been leading in some of the key areas of success, particularly pioneering new therapies. So new gene therapies, so first in human for development of ADA-SCID gene therapies, first in human trials for CD19 CAR T cells, as well as a number of other areas. And that links strongly to our translational activity, where there's been a number of spin-outs from UCL being led by a number of the investigators. And 2 of the projects being offered this year, those led by Dr. Martin Pule and Dr. Claire Roddy, who both previously been involved in leading some of the spin-out activity, particularly the work that's been led by Autolus. Next slide, please. And my final slide just really is to touch on highlighting that, I think these projects capture the spirit that UCL has, which is we're ambitious, we're innovators, and we want to be visionary trendsetters, and we want to be disruptive thinkers. So please do consider applying to the UCL projects. I'll now pass back to James. I don't know if we have Kath on the line. Oh, Kath. Fantastic to have you. I'll pass over to Kath from University of Oxford.
Hello, thanks, Neelam. I'm Kath Green. I am one of the Oxford representatives on this ABViP programme. It's very nice to be here with you all this evening. I head clinical biomanufacturing at the University of Oxford, so I'm involved in the manufacture of medicines for first in human trials. And so the ABViP programme sits within the University of Oxford's Medical Sciences division, and Medical Sciences division is the home for many of the different departments that do medical research and basic bioscientific research. I'm housed in the Nuffield Department of Medicine, but the DPhil programs that we offer as part of the ABViP program will be housed across the Medical Sciences division, not only in the department that I am in. Next slide, please, Neelam. So the Medical Sciences division has its own doctoral training centre, and we sit this programme within that to enable the students that are at Oxford on this course to participate in training centre activities. So within that, we have PhD students that are funded by a wide variety of routes. They might be funded by external programmes such as the Wellcome Trust or Cancer Research UK. They may have internal funding from divisions and departments. So a broad range of students participate in training that operates across our doctoral training centre, and you can avail yourself of any training programmes that a doctoral training centre provides, and they cross the range from computer skills, presentation skills, dealing with the public and how to write a thesis, and maybe even how to manage your supervisor. Next slide, please, Nina. And at college, we have this unique setup where every student belongs to a college as well as a department, and your college is your home from home. You may live in accommodation owned by your college, you may eat meals at your college, and the joy of a college is not only are they quite often very beautiful places to be, but they have a lot of extracurricular activities, be that music, they often have choirs in ancient chapels, be that sports teams, or just a way to meet people that are not medical science students and therefore have broad range of interests. I think those of us who attend Oxford or Cambridge go away with friends from across the fields of not only science but also the humanities, and that's a very valuable aspect to the life that you have when you're in an Oxford college. But if you don't know which college you would want to apply to as part of this programme, don't worry. Neelam can help you with either deciding on a college, or we have a system where students can be allocated to colleges. So if you're not sure and haven't visited Oxford, don't let that put you off. We can help you with finding a college that's right for you. Neelam, next slide. So as I said, we have skills training across the Medical Sciences division. So not only do the students that come to Oxford benefit from the training provided by the ABViP programme, but they also have access to the wide range of courses offered by Oxford in the same way that UCL have some UCL specific courses offered to their students as well. Next slide, please, Nina. So we pride ourselves in Oxford on entrepreneurship and equipping our students for their future, whether that lies in academia or not. And I think we've shown over the work that we did in COVID that Oxford can lead the way in innovation and entrepreneurship. And so we feed our students into contacts with our tech transfer teams, with our innovation teams, manage any intellectual property that arises via this programme or in any other routes that you take. I think Oxford is a thriving ecosystem, obviously with Oxford Biomedica on our doorstep and many other local companies that we work together with. So there are opportunities for growth post this PhD programme. Next slide, Neelam, please. I'm done.
Yep, so thanks, Kath. Just going to quickly run through the list of projects. So there are 8 projects that are open for student applications. They're open now, and you can find this information available on our website, the UCL website, and the University of Oxford website. So I'm not going to read them out because the next series of slides are pre-recorded slides provided for each, giving a bit of background for each of the projects. So thanks very much, Neelam, and I'll go offline.
My name is Dr. Susan Morris, and I'll be jointly supervising the DPhil project titled Characterization of Adenovirus Vector Vaccine Particles for Improved Antigen Delivery, alongside Professor Dame Sarah Gilbert. The Gilbert Group is part of the Pandemic Sciences Institute at the University of Oxford. Within the Institute, multiple groups are working to bring together a wealth of knowledge and expertise in infectious disease, immunology, vaccinology, and global health. As a multidisciplinary collaborative centre, it works with global partners to confront the challenge of epidemic and pandemic infectious diseases. Professor Sarah Gilbert is leading a research team to generate vaccines against emerging pathogens. Our group played a pivotal role during the COVID-19 pandemic through the development of the Oxford AstraZeneca vaccine, and continues to lead on vaccine development and testing for many emerging pathogens. The Gilbert Group's activities cover all aspects of vaccinology, from vaccine design, vector development, rapid GMP manufacturing of vaccines, vaccine delivery, and development of clinical trial assays. Replication in competent human simian adenoviral vectors have been extensively used as a delivery vehicle for infectious disease antigens. These vectors infect replicating and non-replicating cells, have a broad tissue tropism, and induce a strong humoral and cellular immune response. However, a significant percentage of adenoviral particles in any adenovirus vaccine preparation are non-infectious. Typically, only 1% of the total number of viral particles in a preparation are infectious. That is to say that the particle-to-infectivity ratio is 100 to 1, although this is adenovirus subtype dependent and vaccine batch dependent. Only those vectors that infect a cell and express the antigen elicit an immune response. The non-infectious adenovirus vectors carry the genomic DNA containing the transgene cassette, and therefore have the potential to deliver the antigen into cells. If we can convert these non-infectious adenovirus particles into effective delivery vehicles, this would result in a reduction in manufacturing costs and lower, more potent vaccine doses, which translates to cheaper, more affordable vaccines. We currently do not understand the mechanism underlying the non-infectious property of the non-infectious viral particles. The limitation of infectivity may be one or more steps within the viral vector manufacturing and delivery process. This could be receptor binding, endosomal escape, genomic DNA transportation to the nucleus, maturation of particles, or stability of mature particles during purification. Therefore, the aim of this DPhil research project is to study the physical characteristics of the different adenoviral vector virus particles using techniques such as flow virometry, cryo-electron microscopy, and atomic force microscopy, and to improve the collective understanding of viral particle cellular interactions involved in vector production using confocal microscopy and cell binding assays. This information will be used to engineer either adenovirus vectors or producer cell lines, or to modify the manufacturing process to reduce batch particle to infectivity ratios, with the overall objective to improve scale-up and production of adenoviral vectors for vaccination and therapeutic applications. It is hoped that this work can lead to the development of more easily manufactured, cost-effective and immunogenic vaccines in the future, which can be accessed globally and equitably.
Hi everyone, so my name is Omer Dushek, and together with Anton van der Merwe, we run the Molecular Immunology Group at the Sir William Dunn School of Pathology at the University of Oxford. We have a diverse set of scientists that are funded by the Medical Research Council as well as the Wellcome Trust, and our primary research interest is to understand how very important white blood cells called T cells recognize antigen on the surfaces of antigen-presenting cells. This recognition event is absolutely critical for recognizing infections and fighting cancers, and we know that it takes place based on receptor ligand interactions at the cell-cell interface. What our labs have been particularly focused on in the last 10 years is to understand how signaling from a variety of surface receptors regulates a variety of different T-cell responses, including T-cell proliferation. In this collaborative project with Oxford Biomedica, we will use our expertise in T-cell signaling to design vectors that can induce transient cell activation in order to improve the delivery of therapeutic antigen receptors. These CAR T-cells and TCR T-cell therapies have the potential to revolutionize cancer treatment. They work by taking T-cells from cancer patients, inducing them to proliferate in vitro, and then at the same time stably transducing a chimeric receptor before being refused into the patient, generating a population of T-cells that can now specifically recognize the cancer cells. Although this therapy is promising, its major limitation is that the complicated logistics of the cell therapy means that the cost is extremely high, and therefore only a small subset of patients benefit. We would like to be able to expand this therapy to a much larger subset of patients by dramatically reducing the cost by injecting patients with the viral platforms directly to transduce the cells in vivo. A major limitation of this at the moment is that the cells in vivo are not proliferating. And so unlike the in vitro experiment where proliferation is induced, the stable transduction in vivo is inefficient. In this project, we will identify genes that can be modified in order to induce the transient proliferation of T cells so that the viral can stably integrate. We will use primary human T cells and synthetic biology approaches to identify molecules that can be expressed in T cells to induce their transient activation. In this way, we will use our expertise in signal transduction as well as the expertise of Oxford Biomedica to produce these vectors to dramatically improve the accessibility of these really promising cancer therapies.
Hi, my name is Professor Steve Hyde.
And my name is Jakob Helmer.
I would like to tell you about our project where we're trying to develop humanized retroelements. I'm the group leader of the Gene Medicine Research Group at the John Rankin Hospital at the University of Oxford. We have a nice array of postdocs, students, and research assistants with a long history of developing gene therapy vectors, particularly lentiviral vectors. This particular project is all around the delivery of genome editing toolkits.
Yeah, so there's currently a lot of issues with the way CRISPR therapeutics are delivered using AAV and lentivirus, and that is in particular because both of them have a limited packaging capacity. There's a risk of unwanted insertional mutagenesis, and both of them tend to give prolonged or lifelong expression, leading to accumulating levels of genotoxicity. All of these issues are in essence related to the vector genome, so we hypothesized that we could engineer a particle that contains ribonucleoprotein complexes instead of vector genomes. And in order to do so, we developed what we refer to as lentivirus-derived nanoparticles. So basically, it is a lentivirus where the vector genome has been replaced with a Cas9 RNP complex, a base editor or a prime editor, and we can use these systems to make a targeted knockout or install a precise edit at our target loci. So lentivirus-derived nanoparticles comes with a number of benefits. First of all, they are not limited by the conventional packaging capacity. There's no risk of insertional mutagenesis because they don't have a vector genome. And the turnover of the therapeutic payload is very short, which means that we generally see reduced levels of genotoxicity compared to other vehicles. And as this system is based on ABViP, it is compatible with both lentiviral serotyping and it can transduce both dividing and non-dividing cells. However, one thing we have not addressed yet is the potential for host immune response towards the ABViP-derived components of the lentiviral platform. And in order to address that, we have carried a analysis in the human genome, and we identified a lots of domesticated lentiviruses that contains the GAG and GAG-POL domains. And we have been specifically looking for human endogenous retroviruses that share the domain structure with the LVNP, so we could easily adopt them for protein delivery instead of vector genomes. And in order to do so, we identified, for instance, PEG10 that is able to vary Cas9 RNP complexes and disrupt target genes in various cell types. So the main objective of this proposal is to harness additional human endogenous retroviruses for delivery to different organs. And in order to make them completely humanized, we will also recompose human endogenous fusion genes. And of course, we will evaluate the immunogenicity using these particles in humanized mice and eventually target recurrent genetic defects. So the overall aim is to develop a new generation of vehicles for potent and safe delivery of CRISPR therapeutics.
We look forward to receiving your applications.
Hello. This project will look at how different viral vectors, which are often used in vaccine development, activate the innate and subsequently the adaptive immune system. The supervisory team consists of myself, Anita Militic at the Jenner Institute in Oxford, Jan Ravinkel at the Weatherall Institute for Molecular Medicine here at Oxford, who is expert on innate signaling, and Thomas Hanke, also at the Jenner Institute like myself, who is expert on HIV vaccines. This diagram here shows the approaches that we plan to take to study how viral vectors activate the immune system. So we will use mouse model systems where systemically we can vaccinate the mice with the viral vectors and analyze a range of readouts. We can also look at in vitro systems where we can derive bone marrow derived dendritic cells and macrophages from mice, wild type as well as knockout, to study the innate signaling on a cellular single cell level. And also we have a novel system of human lymph nodes where we can take healthy human lymph nodes from surgery, slice them into 300 micron sections, and then stimulate them with viruses ex vivo to study how the human immune system, lymphatic system, responds to these viruses. So here is a little bit more detail about the possible approaches that we might be taking. So we can use fluorescently labeled viral vectors by inserting, for example, green fluorescent protein, which can be used as a signaling molecule or a reporter molecule to see which cells and which organs are picking up the viruses. So this way we can see where the virus is ending up in the mouse body, and also which cells are picking up the virus, and then study the spatial and the kinetic distribution of the viruses, of each individual type of virus. The viruses we plan to study adenovirus, modified vaccinia ankara or MVA, parainfluenza virus 5, which is used in clinical trials against HIV, and also lentiviruses. So we plan to really look across different viral platforms to somewhat systemically analyze how they differ in the pathways that they activate. So apart from looking at viral tropism using fluorescent labeling, we would also, or using an in vitro system, look at signaling pathways that are activated by these individual viruses. For example, at the various inflammatory or cytokine-inducing pathways. And readouts there would be various cytokines as well as cell death, for example. And then also looking systemically in mice, we will analyze the adaptive immune response to the individual platforms by looking at B-cell and T-cell immunity. So although the main focus of the project is how different viral vectors induce T-cell immunity, as they're one of the few platforms that specifically enhances T-cell immunity. For completeness, we would also look at the antibody responses to these viruses and try and really understand a bit more about how different innate signaling pathways lead to specific adaptive responses, which is one of the focal points of our group, trying to understand the interplay between innate and adaptive immunity in the context of vaccination, and more specifically in the context of viral vectored vaccine platforms. That's all I had to say. Thank you very much for your attention.
Hello, this is a short presentation on our PhD studentship, which is BBSRC funded, and is all to do with taking a chemical biology approach to understanding these innate defenses of T cells to lentiviruses. So what does that mean? So lentiviruses, which are modified forms of HIV, are used to carry genes into T cells. Probably heard of CAR T cells, which is one of the main examples where they're employed. So these are great agents for the treatment of some cancers. And what we do in my group is to take chemical approaches to this problem. And I've just given an illustration of some of our chemistry here on the left. You can see we just do a very simple amide coupling, and then we do one of these Grubbs metathesis chemistries. This is a beautiful piece of chemistry because it allows us to essentially take a natural product, cyclosporine in this case, and to join on another complicated molecule to make this really very large structure, which is able to specifically remove particular proteins from cells. And it allows us to do all sorts of things. It allows us to study mechanisms. It allows us to discover mechanisms. So I'll just illustrate that on this slide. And what you can see here is the mechanism by which these molecules work, which is a PROTAC mechanism. I don't have time to describe that. You can be sure you can read the process. So here's the molecule here, and what it does is it degrades the protein in cells. And so what you've got at the top is a proteomics study. And here you can see that the proteins that are showing up outside the line are those that the compound induces degradation. So it's pulling out its targets here, some of these cyclophilin proteins. So this is a very nice specific way to study mechanism in T cells. This study was in the model T cell. And so we can use this particular idea to study mechanisms in T cells and hopefully produce agents that are going to enhance the transduction process, all leading to a better understanding of T cells and how they work and how we might improve these kind of gene therapy techniques in the future.
In this project, we aim to study heterogeneous viral vector population both experimentally and through modeling. The project will be supervised by Dr. Frank Bagans and Dr. Stephen Goldrick from the Department of Biochemical Engineering at UCL. Frank Bagans is an associate professor in fermentation and cell culture, and his research is focused on modeling, engineering, and scale-up and scale-down of fermentation and cell culture processes. Stephen Goldrick is a lecturer in digital bioprocess engineering, specializing in the application of advanced data analytics and mathematical modeling to the biotechnology sector. The study of viruses for applications in cell and gene therapy and viral vaccines is one of the most rapidly growing areas in biotechnology. However, current efforts at characterizing viral populations have been limited by insufficient attention to their heterogeneous nature. The aim of this project is to characterize viral vector heterogeneity by accounting explicitly for the distribution of sizes, shapes, and functional properties exhibited by experimental populations. The primary method for the measurement of viral heterogeneity will be flow cytometry that will be supplemented by other techniques such as particle sizing using multi-angle dynamic light scattering. Quantitative tools will be used to characterize viral distributions, including mechanistic and data-driven approaches. These quantitative methods will produce mathematical descriptions of whole viral populations. The outcome of this project is a detailed quantitative understanding of the viral vector population, including vectors with intact genome, those with fragmented or incomplete genome, and empty particles under various operating conditions.
Hello everyone, my name is Leila and I will be presenting today an overview of one of the PhD projects in Dr. Roddy's lab titled Engineering Lentiviral Vectors for In Vivo CAR T-Cell Manufacturing. So to start with an overview of autologous CAR T-cell therapy as shown in the diagram on the left-hand side, patient T-cells are isolated from a leukapheresis followed by activation and transduction using viral vectors to produce CAR T-cells. For the purpose of this project, we will be focusing on lentiviral vectors. Transduced cells are then expanded, and once the therapeutic dose is reached, cells are injected back into the patient to hunt down and kill cancer cells. Now, taking a closer look at steps 1 to 4, a schematic diagram is presented in B on the right-hand side with an emphasis on the production of lentiviral vectors. So this is achieved by first transiently transfecting HEG293 T cells using 4 plasmids. To go through them quickly, the first one in red is the envelope glycoprotein, which is used to pseudotype the viral particles. Commonly, it's VSVG. Then we have the polyprotein GAGPO in green and the REV protein in yellow. Both of these are HIV-1 proteins. And the fourth plasmid depicted in pink is the transfer vector, which is the plasmid that encodes the therapeutic CAR. Once all the proteins are translated in the 293 T cells, the RNA encoding the CAR is packaged into particles assembled on the cytoplasmic side of the plasma membrane. The envelope protein is acquired through passive incorporation as these particles bud from the cell, producing lentiviral vectors. So we're at step 5. The harvest is then subjected to extensive downstream processing to remove impurities and concentrate the viral titer before these vectors can be used for clinical manufacturing. So purified vectors, step 6 at this point, can then be added to a culture chamber containing previously isolated and activated T cells, leading to the generation of CAR-Ts as we saw in the schematic diagram in A. Now, although this process has completely revolutionized the treatment of certain cancers, its manufacturing remains a major bottleneck for its widespread application. It is incredibly costly, complex, and time-consuming. One way to efficiently tackle this bottleneck is to disrupt the current manufacturing and explore in vivo CAR T-cell manufacturing. And this is what we want to investigate with this project. So the main aims here are first to decrease the cost of CAR T-cells by generating a universal vector product per treatment. Secondly, it's to increase the anti-tumor efficacy of these CAR-Ts by selectively targeting the less differentiated T-cell subset, which is the naive and central memory compartments. So these aims will be tested by generating engineered lentiviral vectors that do not express a viral envelope as seen in the previous slide, but instead they express synthetic proteins that would be passively incorporated on the surface of these particles. So the first one would be single-chain variable fragments against markers expressed on naive and central memory T cells, and a second one would be fusion protein to allow cell entry. So engineering particles using passive incorporation is a very attractive method as it enables the possibility to test a range of different combinations without any genetic alteration of the viral components. So therefore the titer should not be impacted. So these engineered lentis would then have to be extensively purified for in vivo application. And more importantly, they have to demonstrate specificity and selectivity for T-cell transduction in vivo using animal models. And if successful, these vectors can then be used as a universal vector products in patients, which would also ultimately enable quicker treatment timing. So thank you very much for your attention and thanks for your interest in this project, and we look forward to reviewing your applications.
Hello, I am going to talk to you today about a PhD project that is available in the laboratory of Dr. Claire Roddy at the Cancer Institute at UCL. The project will investigate the production of semi-synthetic lentivirus. This project seeks to address some of the limitations of current lentiviral vectors. One of the first problems of lentiviral vectors is that aberrant splicing of the lentiviral genome RNA can occur during production in packaging cells. The production of lentiviral vectors typically consists of transfecting packaging cells with plasmids encoding HIV GAGPol, REV, envelope glycoprotein, and the lentiviral genome. Transcription of the lentiviral genome RNA is driven by a promoter that is upstream of the 5' long-term repeat, LTR. Usually this gives rise to full-length transcripts. However, aberrant splicing of the genome RNA can occur due to the presence of cryptic splice sites in the lentiviral genome RNA. This can result in the production of non-functional lentiviral genomes that can be packaged into lentiviral particles. A second limitation of current lentiviral vectors becomes evident after insertion of the provirus to the host genome. Lentiviral vectors are designed in such a way that transcription of their cargo is driven by an internal promoter, represented as a white arrow in the diagram. But termination of transcription is controlled by a polyadenylation signal sequence located in the 3' LTR. As the polyadenylation signal sequence is relatively weak, transcriptional read-through can occur, leading to transcription of genes downstream of the 3' LTR. This is undesirable and it reduces the safety profile of lentiviral vectors. A third limitation is that the transcript generated from the internal expression cassette may be unstable due to the lack of a 5' intron. Currently, the production of lentiviral vectors in packaging cells precludes the inclusion of 5' intron internal expression cassette because it would be spliced out by the packaging cell splicing machinery. Incorporation of a 5' intron would be beneficial as it would increase the stability of the transcripts generated from the internal expression cassette. We believe that producing semi-synthetic lentivirus may offer a solution to the limitations of current lentiviral packaging systems. The idea would be to produce the lentiviral genome RNA in vitro using an RNA polymerase. This would overcome the problem of aberrant splicing of the lentiviral genome RNA, as there would be no splicing machinery present in the in vitro transcription reaction. It would also enable the re-engineering of the lentiviral genome to include regulatory or stabilizing elements such as 5' intron, represented as an A in the diagram, or a polyadenylation signal sequence, represented as a B. The inclusion of these elements should not only help to stabilize the RNA transcripts expressed from the internal expression cassette containing the cargo, but also increase the safety profile. After generating the lentiviral genome RNAs in vitro, they would then need to be packaged into viral particles. This could be achieved by producing viral-like particles, VLPs, by transfecting packaging cells with GAG-POL, REV, and envelope glycoprotein in the absence of a genome plasmid. The VLPs could then be transiently permeabilized to allow entry of the in vitro transcribed lentiviral genome RNA and the generation of functional lentiviral particles. Thank you for your time.
Great. So we're just going to quickly whiz through a little bit of an overview of the application process, and then we'll be opening for Q&A. So next slide, please, Nina. So in relation to the requirements and information, we aim to have 8 students, so that's 4 each at UCL and at University of Oxford, starting in September, October 2024. To be eligible, you will require a minimal grade of an upper second class UK bachelor's degree in a relevant discipline or an overseas equivalent qualification. In relation to home status, that's considered as UK national, settled status, pre-settled status, or indefinitely leave to remain. That will have to be assessed at the point of application. Only one overseas student will be eligible at each academic institution per cohort. So that's an important point. That's not a specific thing that Oxford Biomedica or the partnership has put in place. It's a requirement, a prerequisite of UKRI funding. So it's not something that we're able to do anything differently. You'd require a CV, a personal statement, and 2 letters of recommendation as part of the application process. At this time, it's also a good idea to inform your referees, as they'll be sent an automatic request to complete them. You can apply to a maximum of 3. So again, this is important. There are 8 projects. Please don't apply to all 8. You can apply to a maximum of 3 projects across the programme with either one or both academic partners. And these will be need to be listed in your cover letter. Next slide, please. So where to apply? The DPhil or PhD will be awarded by the academic institution. So if you're registered at UCL, you'll have your PhD from UCL. If you're registered at Oxford, you'll have your DPhil awarded by University of Oxford. All projects are open to application. Please register your interest with CareerMap or by emailing directly to the ABViP email address. So that's abvip@oxb.com. The deadline for applications is Friday, the 1st of December. So you have a few more weeks at 12 noon. And then there are some links here, and you'll be able to access these a recording of this session later on the university websites for both Oxford and for UCL. Next slide, please. So without further ado, I think that's the end of the presentation phase. We can open it up to Q&A now. So if the rest of the presenters would like to come online, we're happy to take those.
Let's see. We don't have many questions. James, you've answered.
Yeah, we haven't had many
questions. So obviously, well, do you want to go over the ones that you've obviously answered? Just the voice.
Yeah, so I can anonymize one. So a couple of them, I assume not everyone can see these because some of them are private. So yeah. So we did have a question about whether it's advisable to contact the supervisors directly. So that can be done. And at the same time, we'd also, if you'd rather have an introduction provided, you can contact through the ABViP email address.
But it's highly recommended to speak to supervisors beforehand. Yes. They like it. Academics are always happy to talk about their science. They want to hear your ideas. They want to see if that's a good fit. So definitely get in touch. It's always a good thing. That isn't only true for our program. If you're applying for other programs, it's probably also a good idea.
Absolutely. Yeah.
And maybe just find one final point. If you don't receive a response from a supervisor, don't be disheartened. It could be that they are inundated with emails. So please do still put your application in. It won't disadvantage you.
It won't disadvantage you at all. Yep.
Yep. Well, you're saying on the supervisors there, we got a question from David saying, will the cover letters be read by the supervisors?
Of course.
Perfect. There you go, David.
We get the full package. We'll read your CV, we'll read your cover letter or statement of purpose or whatever they're called in each institution. You want to tell us as much about yourselves as possible. You want to tell us how enthusiastic you are about the course, and you want to convince us that you're the right person for this job.
Yeah, perfect. Question from Amy now. Are there any guidelines to what should be outlined in the personal statements or how long they should be?
We have a word limit. I don't know what it is, but I know it's on the form. Neelam, do you know what the word limits are?
No, I don't actually.
It's
not too long is key.
Yeah, and it might be specific to each institution. In response to kind of what should be outlined, I think with any personal statement, obviously you're applying for a research doctoral program, so highlighting any relevant experience you have, any expertise you have, I think importantly why you want to apply for it, and what makes you a strong candidate. Those are the things I think we'll be looking for. We recognize obviously you're not going to have years of experience in the lab, but if you do have any lab experience, do highlight that.
Perfect, thank you. Question from Isa. Do both of our referees have to be academic? This might be difficult if we have left university more than 5 years ago.
If you have left university more than 5 years ago and you can get an academic reference, that's good. But relevant recent references from employers, we need to know about your skill set. We need to know about what it is about you that makes you good at doing a PhD. So those references need to be from serious people, but they don't necessarily have to be academics. People who understand your skill set and can sing your praises is probably what you want.
There you go. And also it's great, Catherine, for you to be here as well. Great for you to come on.
Sorry, I was slightly delayed. I was coming in from Brussels. One of the joys of being a scientist is the chance to travel and meet interesting scientists from across the country and across the world.
There you go. Well, all the questions are coming in now. Here we are. Question from Carla. If we are applying for 2 projects, do we need to upload the same cover letter? Will it be relevant to each project?
Yeah, I think strategically, I mean, if the covering letter is broad enough that it can cover both projects, great. If you actually want to specify something for a particular project which might be relevant, then obviously make it more distinct and keep them different. But I'm sure some of it will be very similar.
Great, thank you. Question from Ines again. Are there any tuition fees for students coming from the EU? If yes, how much would they amount?
So James, you know this one.
Yeah, so my understanding is if you're an overseas student and you are one of the students that gets funded, then that gets funded as part of the package.
Yeah, so just to clarify, So EU students are now classified as overseas students, which wasn't previously the case prior to Brexit. Having said that, there's a slight potential opportunity to be considered a home student if you have settled status. So do check with the respective university as to what your status is. If you are, however, considered to be a European student, it would therefore be an overseas application. And as James mentioned, we can only have one overseas applicant at UCL and one overseas applicant at Oxford University.
Yes, but those would have their fees paid.
Yes,
Correct.
exactly. Yeah, they would have their fees fully paid and they would receive the stipend.
Very nice. There you go, guys. Hope that answers your question there. Question from Stefan. Technical question about the UCL application process. If we apply to both EPSRC times 5
I would say with that question, let's take that offline because that's a very specific question. So Stefan, if you reach out to me privately, and also you can reach out to the UCL admin team because that's to do with other doctoral programs as well that we have.
There you go. Probably the same for Stefan's asked that other one as well.
The one about 3 from Oxford or 3 from UCL. So no, it's 3 across the ABViP programme. So of the 8, so 4 at UCL, 4 at Oxford, please only apply to 3. So a maximum of either 3 at Oxford or 3 at UCL or 2 from one and 1 from the other.
What about Stefan's next question there about how you would handle applications for both?
Again, that's the one that Kasim will cover.
Yeah.
All right. Perfect. Isa, if we're currently in the process of applying for UK citizenship, with being citizenship being granted prior to starting the course, can we be considered as a home student?
There will be rules about what status you have on the day that the degree course starts, and that is best handled by speaking to the admissions team at the university to which you are applying.
Perfect. Roberta says, do we express our interest in a project only in the cover letter?
I think for the Oxford application, there's a place on the application form itself. It certainly doesn't hurt to also put it in the cover letter.
Mm-hmm.
Yep. And likewise for UCL, I think there may be an opportunity in the application form. I think expressing interest wherever the opportunity arises, where appropriate, is a good idea.
Okay, perfect. Is pre-settled status considered home status? No, not from the EU.
I think again, so pre-settled students with pre-settled status would be classified as home students, but always check with the university admissions team at the point of application. Even after application, the fee status is determined locally by each university based on the information that you provide. So one piece of advice is, as you're going through the application process and as your application is being considered, please do respond promptly to any information from the universities about uploading documentation, whatever it might be, because that's what they use to make the decision on the fee status.
Mm-hmm.
There are also information on the internet about that. On gov.uk, I think there's some student pages about what qualifies what and what dates things needed to happen. And you're likely also to find those on application pages of the 2 university websites. So if you have a hunt around, because there's quite a lot of detail in there, it depends on countries and dates and parents of origin. So we can't really cover all of that now, but there's quite a lot of information on the internet that you should be able to find out about which status fits in which box.
Perfect. Thank you, Catherine. Well, what about, just so we can get it on video as well, for final application date for closing? When is that?
1st of December at 12 noon.
There we go. So,
and yes, there is an English proficiency requirement, so you will need to have a level of English which is acceptable to the university. Again, that might be different at the 2 universities. Neelam, do you know if we have different requirements? Do you know what our English language requirements are?
Not off the top of my head, but in the links to where you apply, there's all of the information that you'll need to know before you apply on each of the websites.
Okay, perfect. Well, any final words of advice from you guys for the people attending the webinar for joining, if they would like to join Oxford Biomedica? What advice would you give them?
So my take on it, this is obviously there are lots of choices out there for PhDs. These are well-funded PhDs as part of quite a unique collaborative training partnership between Oxford Biomedica, University of Oxford and UCL. We're obviously building up the cohort, so we now have 16 students on the programme already from cohorts 1 and 2. So this is an opportunity to join and become one of 24. So I think compared to my experience as a PhD student, which was many, many years ago, it's very different. And I think it's actually really reflected on very positively by the students that are on programme right now. And it is pretty competitive. Being fully funded and so on does sort of make it a competitive application process. So really put in your best efforts to apply, and we'd encourage you to.
This is a fantastic programme. We're looking forward to some bright new minds to come and join our teams. So we look forward to seeing as many applications as possible.
Thank you. Who's next?
I think Kath and Jim said everything. We're looking forward to your applications. Please do ensure you submit them on time. We won't be considering late applications. And I think there's a question about interviews. There will be interviews after the application process.
They will be in January. We will shortlist candidates for interview, and we'll have a fairly intensive day of interviews. So yes, we will. The panel and the supervisors will do a shortlisting process, and then we will interview a selection of candidates for interview with the supervisors of the projects and members of the board.
Amazing.
That will be online interviews. You will be doing that online.
There's a question from Amy about the reference deadline. I'm not sure I'm not sure what that means.
So I think this is to get references. I think if there's any admin type questions, they can be followed up afterwards as well. And Neil can give the ABViP email address. Ideally, references should be submitted at the same time as the application.
Mm-hmm. Do you want to pop them in the chat?
There's a system for that. So you put your you put your referees' details into the application form, and then they have to send in the references via the university systems. Certainly at Oxford, I presume that's the same at UCL. So if you're going to nominate somebody to be your referee, you need to tell them that you are doing that, so that they are prepared to get the email from either UCL or Oxford and send in the appropriate reference.
Perfect. Thank you. We'll just get Cassie putting them links into the chat for you guys in the webinar. There we are. Perfect. Well, it is now, we're over time just by a few minutes, but that has been a very insightful webinar. Thank you very much, everyone from Oxford Biomedica, for taking everyone who joined the webinar through this. Remember, you can check it out on our YouTube and our website within the coming days if you would like to watch our lovely faces again. Thank you very much. Hope everyone has a lovely evening.
Have a good evening, everybody.
Thank you very much, everyone.
Bye-bye.
Bye-bye.
Thank you guys.